Well, it's my pleasure to welcome once again Dr. Helen Collins to speak with us this afternoon. Dr. Collins is an assistant professor in medicine in the Center for Cardiometabolic Science, and she has joint appointments or secondary appointments in physiology as well as biochemistry and molecular genetics. She received a BS with honors from Leicester University in the United Kingdom, and then did a Ph.D. in cardiovascular sciences, also at the University of Leicester in 2011. She has published widely from that early work in her career. She then served as a research assistant at the Open University. And from 2012 to 2019, Dr. Collins was a postdoctoral scholar in the Department of Pathology at the University of Alabama. And there she published 14 papers with additional papers stemming from that research once she moved to UofL, where she has been since 2019. She's participated in a number of training opportunities in teaching and mentoring, and she's been very successful. One of her trainees is entering the MD PhD program here at UofL. She's received a number of awards for her research, including the 2022 Division of Environmental Medicine Faculty of the Year, Kentuckiana American Heart Association 2024 Live Fierce Award. She was named an extraordinary reviewer for 2024-25 from Circulation Research and named a star reviewer in 2021 for American Journal of Physiology, Heart and Circulatory Physiology. She's now a consulting editor for AJP Heart. She's on the editorial boards for 7 journals, including one of my favorites, Molecular and Cellular Endocrinology, and has served on a wide range and number of AHA and NIH grant review panels, as well as a number of other review activities. And she, of course, has presented her work at national and international meetings. She's currently serving on 2 PhD committees, mentoring a postdoc, 2 master's students, and has trained 2 NIDDK T35 summer medical students in her lab, which I really appreciate. She also has mentored a number of undergraduate and high school students. She's currently PI on an NIH HL R01 grant, has NIGMS IDEA network-funded proteomics voucher, and is co-investigator on 2 R1s. And of course, she has a number of grants submitted and under review, as well as papers as under review. She has published 31 papers, and she has she did a presentation in South Korea in 2024, so she certainly has presented her work internationally. So it's a real honor and pleasure to have Dr. Collins give us an update on her research today. So thank you.
Thank you for that kind introduction. So I'm going to take us in a different direction to how I've usually pitched this work in this arena before. So these are my disclosures. So for anyone in the audience that's not familiar with me and my work, the mission of my lab is to understand how the female heart adapts in the setting of both physiological and pathological stress. So with respect to physiological stress, we've looked at pregnancy, and with respect to pathological stress, we've looked at the impact of myocardial infarction. So I'll start with the physiological stress. How does the female heart respond to physiological stresses? So, as I mentioned, one physiological stress test that women will undergo is pregnancy. However, in the United States, being pregnant is very dangerous. These are the maternal mortality rates from 2018. And you'll note here that the US leads the pack with 17.4 maternal deaths per 100,000 live births. And this is significantly higher than the rest of the developed world. More recently, these statistics were updated, and you'll see the blue bar here is 2018, which is depicted in this diagram here, of 17.4 deaths per 100,000 live births. However, since then, at 2021, this has increased to 32.9 deaths per 100,000 live births. There is a slight caveat to this 2021 data. There was some overestimations in reporting of maternal mortality. And this is reflected in this scheme here, where we see the new stats in 2022 with the new collection regime indicate that this maternal mortality rate is not as dramatic as it was in 2021 and more comparable to the rates seen in 2020. However, regardless of this, these maternal mortality rates are still significantly higher than the rest of the world in the US, and any maternal death is one death too many. So there are several significant factors that contribute to maternal mortality. Around 15% of all maternal mortality is comprised of cardiovascular disease. And there are several risk factors that contribute to this cardiovascular disease during pregnancy. One, which has arisen in the last decade, is advanced maternal age. This scheme here shows that women 40 years or over have a significantly higher maternal mortality rate in comparison to women between 25 to 39 or 25 and under. And also with the newly revised data collection methods, this trend is still recapitulated. Also, multiple pregnancies are significant maternal disease risk factors. And with respect to gravidity and parity and multiple pregnancies, I am from the Division of Environmental Medicine, so it's important for me to state that smoking and vaping and exposures to toxicants and pollutants are also significant maternal cardiovascular disease risk factors. Also, several factors known to impact systemic metabolism are known to also be risk factors for maternal cardiovascular disease, including obesity, poor nutrition, both type 1 and type 2 diabetes, hyperlipidemia, and something that we're increasingly interested in the lab right now is sleep disordered breathing and sleep disturbance during pregnancy. And then also several risk factors for maternal cardiovascular disease are factors which arise during pregnancy or as a result of pregnancy, such as gestational hypertension, preeclampsia, gestational diabetes, preterm birth, and then also the physiological action of lactation, which is more apparent in peripartum cardiomyopathy, where lactation has been proposed to be a significant player in the pathology there with the inappropriate cleavage of prolactin, vicotepsin enzymes. And then also having C-section is a significant risk factor. It's important for us to appreciate these risk factors and appreciate maternal cardiovascular disease because it's been shown that maternal cardiovascular disease shapes the long-term cardiovascular disease risk of a woman. This data here in JAHA from 2022 shows women between the age of 25 and 35. However, this study also included women under 25 and over 35. And it was shown, irrespective of age, that the presence of a cardiovascular complication during pregnancy increased both all-cause mortality and cardiovascular mortality. So this shows us that the presence of a cardiovascular disease during pregnancy will shape the cardiovascular disease risk in future female life, and also it tells us that we have to do a lot more to study this. But before we do so, we need to understand what happens during normal pregnancies before we can understand the specific pathways that go awry with maternal cardiovascular disease. So what are the normal physiological changes that occur during pregnancy? There are several changes that occur within maternal systemic and cardiovascular physiology during pregnancy. Specifically, these include increases in cardiac output mediated through increases in stroke volume and heart rate, increases in blood volume, neurohumoral changes, Also, vascular changes, including the uterine artery, increase in uterine artery blood flow and remodeling, increase in vascular compliance, a reduction in total peripheral resistance. And most relevant to our work is the systemic and cardiovascular metabolic changes and the development of pregnancy-associated cardiovascular growth. So it's important to note that during pregnancy, the heart does grow in size. However, this is different to what you would observe during pathological remodeling. Pregnancy is defined as a physiological hypertrophy, and this is because during pregnancy, cardiac function is maintained, or in some cases, slightly depressed during late pregnancy, which is not the case with pathological remodeling. Pregnancy-associated hypertrophy is thought to be reversible, and this is also unlike pathological remodeling. With pregnancy, there's minimal fetal gene induction, relatively no fibrosis during a normal pregnancy, and normal changes in capillary density and angiogenesis, which are also unlike pathological remodeling. And although there are several pathways that are evoked in both scenarios, they are significantly different from one another. So in our lab, we've studied some of the processes that occur with physiological cardiac hypertrophy during pregnancy. And in order to do this, we've looked at the FEB NJ mouse strain at these 4 groups. We have a non-pregnant diestrus group, a mid-pregnant group, which reflects day 8 of pregnancy within this strain. We have a late pregnant group, which reflects day 16 of pregnancy. And then we have, for the purposes of this slide, a 1-week postpartum group in which mice are able to maintain their litters and lactate. And so what we've shown so far is that during pregnancy, heart size increases during all stages of pregnancy and postpartum. And this, at least at the postpartum time point, is associated with the development of a concentric hypertrophy. However, studies are ongoing in the lab to investigate the nature of this hypertrophic response in more detail, given the increase in overall cardiac size at late pregnancy without the change in myocyte cross-sectional area, which does not pick up changes in eccentric hypertrophy. So additional analyses are underway right now. We also find that with this pregnancy studies that we don't observe any significant changes in cardiac function as assessed through echocardiography and through ejection fraction measurements. However, we do see those characteristic increases in end diastolic volume and systolic volume, and an increase in cardiac output mediated through increase in stroke volume. And also, in line with the predetermined criteria for physiological hypertrophy, we don't observe any changes in fibrosis at any time point during pregnancy and post-birth, and we don't see any differences in fetal gene expression. So with respect to this specific slide, these are gravimetric sizes here. This is from histology, but we also do have left ventricular mass corrected measurements from echocardiography too.
So the cells are larger, is that what happens?
So basically, we're seeing an increase in cardiomyocyte cross-sectional area post-birth, which is indicative of a concentric hypertrophy. We do see that the heart size is overall larger at late pregnancy. However, the myocyte cross-sectional area measurements that we do only pick up cross-sectional area. They don't pick up myocyte length, which is indicative of eccentric hypertrophy, which is what we think is occurring at late pregnancy. So we need to do those studies now where we isolate cardiomyocyte and look at cardiomyocyte length. It's very highly unlikely to be more cardiomyocytes there. Yeah, so I think it's a change in.
You would think that the heart, like when an athlete is something, the heart pump harder.
Yeah, we do see an increase in cardiac output, but yeah. So yeah, for sure. So we, having established our pregnancy induced cardiac growth model, we next wanted to know the mechanisms that contribute to this growth. So we performed a triomics analysis, and we first looked at the transcriptome using bulk RNA-seq, and we interrogated our 4 groups. And you can see from this heat map of the top 50 changed transcripts, there are significant changes in going from each group. But what's important to note that at mid-pregnancy, there's increases in transcripts associated with cell cycle, cell proliferation, and angiogenesis. If we skip late pregnancy for just a second and move on to post-birth, there's a significant increase in transcripts associated with the extracellular matrix and the fibroblast compartment. And we are actively now interrogating both this mid-pregnancy changes and post-birth changes with more detailed analyses with respect specifically to the post-birth time point, where we're now going to look at extracellular matrix changes in the heart during pregnancy. However, we were most interested in this late pregnancy time point, at which we see an increase in transcripts associated with metabolism. Specifically here, we do see an increase in PDK4 expression, which is important to note because previously Zoltana Rainey's lab has shown that PDK4 expression is also increased in the heart at late pregnancy, and that if you block the activity of PDK4, this actually inhibits cardiac growth. So this was a significant corroborating piece of data that aligns with their study. So perhaps this is a significant player with what we're seeing. So in order to validate some of these transcriptomics data, we next validated that at the protein level using immunoblot analysis. And what we see is comparable to our transcriptomics analysis, that we have an increase in PDK4 protein expression at late pregnancy. We also see a reduction in the phosphorylation of PK2, and thereby a reduction in phosphorylation is indicative of reduction in phosphorylation. And also in our transcriptomics analysis, we also found that the ketone body enzyme beta-hydroxybutyrate dehydrogenase 1 was significantly increased at all stages of pregnancy and post-birth. So we also validated that at the protein level, and we were able to show that BDH1 protein expression was significantly increased in the heart at all stages of pregnancy and post-birth, and followed an expression comparable to the arc of cardiac growth that we observed. So what we think is happening is that we see a reduction in glucose oxidation at the level of both PFK and PDH, and this might drive some of these glucose derived carbons into these pathways here, which are ancillary biosynthetic pathways of glucose metabolism, which are pathways in which they're known to provide building blocks necessary to facilitate cardiac growth. And whilst that's occurring, we think that ketone bodies acting via BDH1 maintain metabolism. So in order to test this hypothesis, we next performed a bulk metabolomics analysis to determine whether any of the metabolites associated with these ancillary biosynthetic pathways were changed. So this is our metabolomics analysis, and these are, I believe, the top 75 changed metabolites, whereby red indicates high abundance and blue indicates low abundance. And what you'll see is there's minimal changes here between non-pregnant and mid-pregnant hearts, with the bulk of the changes occurring during late pregnancy and post-birth. If we look at late pregnancy, this is associated with changes in metabolites associated with amino acid metabolism. Specifically, we found that the urea cycle associated metabolite homoarginine was significantly increased in late pregnant hearts. So that is now an area of active investigation in the lab to determine its importance in the maternal heart. In addition to this, we found several other amino acid metabolites increased in the maternal heart at late pregnancy. In addition, we found several fatty acid metabolites, including several sphingomyelins and long-chain fatty acids, increased in the late pregnant heart.
During pregnancy and late pregnancy, one might think, I mean, I get that you're looking at the metabolites in the heart, but somehow those metabolites, have to come, there's exchange between other tissue compartments. So wouldn't you expect there to be more glucose metabolism, like metabolites are being shunted into the uterus to feed the growing fetuses in mice, many of them. And then is there a difference in metabolism in the heart if the mice have their offspring with them versus if they have regression of the mammary gland and are not lactating?
So I can answer that second question in a few slides' time.
Okay.
That first question, yes, there is definitely changes in systemic glucose metabolism whereby shunted towards the fetus too. But there's also differences in substrate availability in circulation during pregnancy. And actually, what is of particular interest to us is the cardiohepatic axis during pregnancy, whereby there's a lot of metabolites produced in the liver that are in circulation and potentially impacting the heart too.
Butyrate.
Yeah, and also homoarginine is produced in liver too. So that was our late pregnancy synopsis. And so when we move to post-birth, we see once again several amino acid metabolites that increased in the postpartum heart. Once again, we see urea cycle associated metabolites increased. And this time we also note that polyamine metabolites increased. And this is a significant finding of interest because previously, polyamine metabolites have been identified and associated with hypertrophy, but mostly in the pathological setting. So it will be interesting to determine their importance in the context of normal pregnancy-induced cardiac remodeling. Yes, this is bulk metabolomics from the heart. Yeah. And so we also observe increases in nucleotide metabolites in the heart post-birth and also glycophospholipid metabolites. So noting the presence of all of these free metabolite classes in abundance in the post-birth and late pregnant hearts indicates to us that there is increased abundance of metabolites associated with ancillary biosynthetic pathways of glucose metabolism. However, because this is a bulk metabolomics analysis, it only gives us a snapshot of what's happening at a given time, and it doesn't give us any indications of what's happening with respect to flux or movement of metabolites into specific pathways. So in order to do this, to investigate flux more so, we performed stable isotope resolved metabolomics and deep network tracing. And in order for us to do this, we gave mice in these 4 groups a liquid diet containing C13 labeled glucose, and we provided this for 18 hours, which has been shown previously to label metabolites of ancillary biosynthetic pathways of glucose metabolism. After which the hearts are freeze clamped and sent for stable isotope resolved metabolomics and deep network tracing. And essentially what we find is increased incorporation of glucose-derived carbon into these ancillary biosynthetic pathways of glucose metabolism, whereby we do see a whole host of many other changes. This is just representative changes that I'm showing here. However, we do see increased movement of glucose-derived carbons into nucleotide production, as evidenced by this increase in enrichment into UMP in this postpartum time point. We also see increased flux into membrane expansion, as evidenced by this increasing glycophosphoglycerol enrichment, and then increased incorporation into amino acid pathways, as shown by this representative L-methionine trace. However, I mentioned that we are looking at the impact of lactation. So that's where we do include this extra group here of post-birth, what we refer to as post-birth non-lactation. And so we did this because in some preliminary studies, we found that lactation has a profound effect on pregnancy-induced cardiac growth. So we have our 4 groups that we've shown previously, but we have a comparable group to this post-birth group here, which is the post-birth non-lactation group, whereby immediately after birth, we remove the pups to reduce lactation. And what we see with respect to body weight is that removal of the pups increases body weight in these post-birth non-lactation mice to a weight comparable with mid-pregnant mice. And then if we look at the heart at this in these same mice, the heart size dramatically decreases to a size comparable with late pregnancy. So it has a profound effect on cardiac remodeling. So we wanted to, because of the expense and the lack of knowledge of metabolic changes in the maternal heart, we also included this group in our stable isotope resolved metabolomic studies. And when we did that, we find reversal of many of these changes. So we find that incorporation into nucleotide production is reduced. We find that incorporation into membrane expansion is reduced, and amino acid metabolism is reduced.
Put the headphones on the computer.
I'll keep moving. I mean, if they want to ask a question, they'll speak up again, I guess. So we find that lactation has a profound effect on the maternal heart and the maternal remodeling response. So we found, because I suggested that we performed a triomics analysis, we also looked at the cardiac proteome. And this is a heat map. Once again, I think this is the top 75 changed proteins in the maternal heart. Once again, you'll note that there's minimal changes between non-pregnant and mid-pregnant hearts, with many of the changes occurring during late pregnancy and post-birth. Specifically, during these 2 time points, which are associated with maximal cardiac growth, we observe significantly lower expression of mitochondrial complex subunit proteins, specifically cytochrome C oxidase and NADH ubiquinone oxidoreductase subunits, several histones, and several contractile proteins. So with this in mind, and our initial theory that ketone bodies maintain mitochondrial metabolism when glucose is being shunted towards biosynthetic pathways, we next wanted to determine whether this specific change here had an impact in cardiac mitochondrial function. How to mute people. Anyway, so in order to do this, we isolated mitochondria from our 4 main time points. Unfortunately, we were unable to include the post-birth non-lactation group in this study, but in our future studies, we intend to include that with our transgenic model. So we provided isolated mitochondria, several different substrate mixtures to assess. We provided a carbohydrate-rich substrate of pyruvate malate, amino acid-rich substrate, glutamate malate, fatty acid, octanol-carnitine malate, and also succinate rotenone. And so we did that, and then we subjected the isolated mitochondria to mitochondrial function analysis using extracellular flux analyzer. And what we find is when we provide a carbohydrate-rich substrate or an amino acid-rich substrate, the mitochondria at the post-birth time point are able to avidly utilize these substrates in a highly efficient manner. We also did not observe any differences, unfortunately, with the octanol-carnitine malate and the succinate-rotenone, but this suggests to us that at the postpartum time point, these mitochondria are able to use these carbohydrate and amino acid sources efficiently. So as I mentioned earlier, that we think that ketone body metabolism is playing a significant role in maintaining metabolism whilst glucose is going towards these ancillary biosynthetic pathways. This is also comes from the fact that we know that changes in ketone body metabolism contribute to metabolic remodeling in the heart. Albeit many of these articles here are in the context of heart failure. However, it's important to note that the heart can use ketone bodies as a metabolic stress defense, and this has been shown by several studies involving mice lacking BDH1 or overexpressing BDH1. So it indicates to us that ketone body metabolism has a significant role in metabolic remodeling in the heart. It's also important to note that during pregnancy, circulating levels of ketone bodies are increased. Ketone bodies specifically are produced in the liver through the action of BDH1, where acetoacetate is converted to beta-hydroxybutyrate. And this is then utilized by extrahepatic organs, such as the heart, for energy metabolism. And so both beta-hydroxybutyrate and also acetoacetate, which are the top two ketone bodies, are significantly increased in circulation during pregnancy. And we've also found that specifically ketone bodies are increased in circulation during late pregnancy in our studies, and this is associated with a reduction in glucose levels too at the same time point. But the role of ketone body metabolism during pregnancy is largely unknown. So with that in mind, we also, in our isolated mitochondrial experiments, subjected mitochondria to the main ketone body, 3-hydroxybutyrate, and we were able to find that the postpartum mitochondria are able to avidly utilize 3-hydroxybutyrate. However, in comparison to the carbohydrate and amino acid derived sources, this is much less efficient. Also, during these studies, we saved some of the isolated mitochondria to confirm at the protein level specific changes in ketone body machinery and metabolic machinery. And we find that in line with this data, that mitochondrial expression of BDH1 at the postpartum time point is significantly increased. And we also find that in line with these observations that PDK4 expression is significantly decreased. So it's mostly beta-hydroxybutyrate that's increased. There is a increase in acetoacetate too. There are thoughts that both will have differing impacts on extrahepatic tissues. However, some of those studies are still under investigation right now, but the predominant ketone body is beta-hydroxybutyrate.
Any of those have no effect on the myocyte, the parasite hypertrophy?
So there's conflicting evidence with respect to hypertrophy. There has been a study by Dr. Sadashima's lab where they treated isolated cardiomyocytes with beta-hydroxybutyrate, and they found that in the setting of phenylephrine stress, and they found that it actually protected against hypertrophy. But what we think is that it protects against adverse remodeling. But that's something that's still under investigation right now. Also, it's important to note that ketone bodies have also been shown to have a significant impact on endothelial cells as well. And also in recent studies from our center, they significantly impact myocardial blood flow. So what I've shown, at least during pregnancy, is that the heart increases in size, and that at mid-pregnancy, this is associated with increases in transcripts associated with angiogenesis, cell cycle, and cell proliferation, and an increase in the expression of BDH1. By late pregnancy, this is the heart is further increased in size, and this is associated with increases in transcripts associated with metabolism, and an increase in PDK4, a decrease in PFK2 associated with a reduction in glucose catabolism, and an increase in BDH1 expression. By post-birth, we begin to see increases in extracellular matrix transcripts, some of the reversal of metabolic changes, namely a decrease in PDK4 expression, and an increase in the ability of mitochondria from these hearts to respire with ketone bodies. And then with both of these time points associated with maximal cardiac growth, we see an increase in the abundance of metabolites associated with ancillary biosynthetic pathways of glucose metabolism and also ketone bodies. So some small studies that we've been doing right now are those which have tried to determine if this cardiac growth response is reversible. And so for this, we've given mice one pregnancy lactation cycle and aged them to 1 year. And then once we have done this, we've extracted the hearts and looked to see if the heart had returned to a non-pregnant-like state. And actually, even a year out, the hearts of the mice that have had a pregnancy are larger than those that have not. This is primarily, it's a small N number, it's a small pilot, but most of the significance seems to be driven by this increase in atrial weight. And then we also see a non-significant change in cardiomyocyte cross-sectional area here. This was important for us to study because this is something that we want to look at in the future to know the impact of a pregnancy lactation cycle on future cardiovascular outcomes when given a secondary stressor later in life. Also, with this in mind, given that several pregnancy-associated cardiovascular diseases and associated with successive pregnancies, and also given the fact that many women have more than one pregnancy, we also examine the effect of that. And what we see with either 1, 2, 3, or 4 pregnancy lactation cycles, that heart size increases with each. And this does not increase further with each subsequent pregnancy, but it is at least associated between 2 and 4 pregnancies with an increase in myocyte cross-sectional area. So this is good preliminary data for us to have moving into more pathological pregnancy studies, but also those looking at the impact of pregnancy and then a secondary stressor later in life. So, as I mentioned, my lab also studies the impact of pathological stresses on female cardiovascular health and resilience. So, how does the female heart respond to pathological stress? So, it's known that heart disease is the leading cause of death in women in the US. And specifically in Kentucky, heart disease is the #1 killer of women, and that's greater than all types of cancer combined. Specifically, Kentucky is number 8 for deaths from heart disease in the US. Unfortunately for women, 50% are misdiagnosed or mistreated after a heart attack. Also, probably is a significant factor in the death rate, but many women delay seeking treatment for heart attack due to the lack of familiarity with the symptoms, which often are not the same symptoms which are seen within men. Also, women are much less likely to receive bystander CPR. And also, women are more likely to die within the first year following an MI than men. However, despite this, women have been underrepresented in cardiovascular clinical trials, where you see here the percentage of women in a randomized clinical trial with this color here, the clear bar being the percentage of women in the population with the disease, and then the number of diseases attributable to each disease. So obviously, this suggests that more needs to be done with respect to recruitment. But why are they underrepresented? It's because women are not aware of the risk of cardiovascular disease, so they're unlikely to sign up for many of these trials. And then also they're unlikely to be recruited into trials because many perceive females to be protected from cardiovascular diseases. There's also a lack of representation, the lack of female investigators in several clinical trials. And then specifically in the ailments that impact premenopausal females, there's very strict birth control requirements for women recruited into those studies. Also, in terms of the basic science component, the use of both sexes in preclinical cardiovascular studies has been historically low. It's only within the last 10 or so years that more females are being used in studies, and more people are looking into sex differences. So it's important to note that women are not just smaller men. We have different cardiovascular changes and different physiology with respect to cardiovascular changes. So we have typically smaller hearts, differences in heart rates, differences in stiffness, especially with exercise and aging, differences in blood volume, differences in perfusion. If you take this down to a cellular level, it's thought that females and male hearts are composed of different percentage total cell numbers with respect to specifically ventricular myocytes and endothelial cells and some other parenchymal cell types too. With this in mind, there are also several cardiovascular diseases that impact each sex differently. With respect to the women, as I mentioned earlier, pregnancy-associated cardiovascular disease is a female-specific risk factor. So it's important for us to study pregnancy to then know, then have a stable foundation for then looking at pregnancy-associated cardiovascular diseases to uncover the pathways contributing so we can then target them later. But with respect to males, they're more likely to have or present with a myocardial infarction. So it's known that in response to an MI, premenopausal females are largely protective and have reduced mortality and reduced rupture rate in comparison to males post-MI. And then also there's a reduced remodeling post-MI in comparison to males. So there are many factors that are thought to contribute to sex differences in the cardiovascular system, differences in hormones, differences in chromosomal contributions, differences, obviously I mentioned earlier, in cell type numbers, but also other genetic factors. So part of the goal of our other studies in the lab is to determine mechanisms that contribute to sex-dependent remodeling of the heart following MI. So what we've done is performed non-reperfused myocardial infarction surgery in males and females, and this time in C57 Black 6J mice. And we find that following MI, heart sizes are much smaller in females versus males. And we also see less chamber dilation in females versus males following MI. And then we also see preserved cardiac function in females versus males following MI. And what we find in our studies is that these changes in post-infarction remodeling are not due to changes in initial infarct size, because when we perform TTC staining, we find that the infarct size 1 day post-MI is comparable between the sexes. So with this in mind, we next then wanted to determine potential factors that contribute to preserved function and reduced adverse remodeling in the female heart. So there's a significant number of changes that occur in the heart following an MI. Several of them are listed on these slides that occur in the remodeling response. And so we were most interested at looking at not only the metabolic remodeling, but also the ECM remodeling, given the changes in left ventricular volumes that we were observing. So we performed some gene ontology analysis, and then when we did this, we found that actually the female hearts had increased enrichment in collagen fibril organization in response to MI in comparison to males. We do also see several immune cell pathways from this gene ontology analysis that we're interested in looking at in the future. So given that we saw this significant increase in collagen fibril organization, we next collaborated with the Moore lab in the center, and we performed polarized light microscopy and curve-line analysis, and we found that there was indications of reduced collagen alignment in the female hearts post-MI. These are active studies that we are still working on, and we're also doing some second harmonic generation imaging to complement this. However, we next performed to try to determine the underlying changes that occur with respect to remodeling. We next performed spatial transcriptomics to have a visual representation of what's happening in the ventricles in the remodeling response. So first of all, we to do this, we take H&E slides, and then we in collaboration with the STC core here at UofL, we perform spatial transcriptomics using the 10x Xenium platform, which has which unfortunately does not have the resolution of the newer platform, which is single cell resolution. But at the time when we were doing this, this was the technology that was available. So what we find is in the infarct zone, this is the infarct zone of the female heart, infarct zone of the male heart. We find in the infarct zone in the female heart, there's several different collagen transcripts increased in this infarct zone of the female heart versus the male heart, COL1A1, COL1A2, and COL3A1. We also see that several collagen cross-linking and stabilization transcripts are also increased in the infarct zone in the female hearts post-MI, which can have a significant impact on the scar properties that we observe between the males and females. So this is something we're interested in following up with. In addition, we see when we did additional bulk metabolomics of male and female hearts in response to MI, we see several changes showing several ECM metabolites, specifically trans-4-hydroxyproline and 5-galactosyl hydroxyl L-lysine. They were significantly increased in male hearts, and this is indicative of collagen degradation products being increased in the male heart compared to the female heart. So it's quite possible that females may exhibit a unique pattern of reparative ECM remodeling compared to males. I didn't show it, but we also see several other ECM-related transcripts increased in the infarct zone of the female heart in comparison to the male heart, which could imply changes in compliance and stiffness, differences in collagen stabilization and cross-linking, and prevention of excessive collagen degradation and controlled remodeling in the female heart. However, these are studies that are still ongoing. So just to quickly end it out, because we know that changes in metabolism are known to contribute to changes in remodeling, we also looked into some of the metabolic changes in the post-infarcted heart. And so in our study in which we looked at the ECM degradation products, for that we did bulk metabolomics. And what we also find when we look at the bulk metabolomics is that not only are there several different pathways that change independent of MI, specifically with the female, once again, similar to some of the changes that we observed in the pregnant heart, we see several sphingomyelins, ceramides, pyrimidines, pantothenate, amino acid, sugar metabolites, and sterols increased. And then when we look at the male hearts, independent of MI, we see increases in metabolites associated with amino acid metabolism, such as histidine metabolites, urea cycle, leucine, isoleucine, and valine, and then also purine and dihydroxy fatty acid metabolites. So we performed some pathway analysis, and where red indicates metabolites decreased in female versus male, and blue corresponds to increased in female versus male. And we see that in female sham hearts, there are several long-chain fatty acids, including oleate, that are reduced versus males, and that post-MI, some of these long-chain fatty acids are more abundant than in the male hearts. We also observed post-MI several monounsaturated and polyunsaturated acyl carnitines are increased in the female hearts versus male hearts. There are a few exceptions. These sex-dependent changes in acyl carnitines were not present in sham hearts. So it's indicating that there are several changes that are occurring in metabolites also that could be associated with the remodeling response that we're interested in. But if I bring you back to this slide, that ketone body metabolism contributes to metabolic remodeling in the heart, it led us to wonder whether it's also contributing to changes between the sexes. And so we performed a experiment in which we can naturally increase ketogenesis by overnight fasting. And when we do this, we see that females have a heightened ketogenic response in comparison to males. And this is also reflected in the compensatory changes in blood glucose. And then in the same MI study, if we look at circulating blood ketone levels are significantly increased in females in response to MI than males. We take this one step further and look more so at some of the ketone body machinery in the hearts in response to MI. You'll note that our favorite enzyme from our pregnancy studies, BDH1, is significantly increased in the area that's dense with surviving cardiomyocytes in comparison to the males. So this is something that we're interested in investigating further and linking back perhaps to some of the ECM changes. So hopefully in this part, I've shown that at least 1 day post-MI, there's no change in infarct size. With day 7 and day 28 after MI, we see increase in cardiac size, which is bigger in the male versus female, increase in chamber dimensions, bigger in the male versus the female, reduction in cardiac function, more so in the male than the female. And this is associated with several increases in ECM components in the infarct zone in the female heart, increases in ECM degradation metabolites in the male heart versus the female heart, and also changes in several metabolites which could afford resilience within the female. So with that in mind, I would like to thank everyone that has worked in my lab or that's collaborated with me over the years here, and that has contributed to both the pregnancy and sex differences related studies. And I'd like to also thank the funding bodies for making this work possible. I'd like to thank you all for listening, and I'd be happy to take any remaining questions, and then I have to throw that up too.
Oh, it's really excellent and intriguing work, and it's really fun to see the story progress. So you have grants submitted to compare sort of a premenopausal model versus a postmenopausal model in which you ovariectomize the mice and see what changes don't or do not occur.
So that's part of the plan. So with every sex differences study, we first have to negotiate the hormonal component, because that estrogen and testosterone have both been shown to have different effects in the cardiovascular system, whereby estrogen has been shown to be more protective. So usually that's what we would do ovarectomies and then rule out any hormonal related changes. For example, the fibrosis component, it's been shown, at least in the ANG2 model, that when you gonadectomize, ovarectomize mice, that the sex differences in the fibrotic response remain, indicating that there's non-hormonal contributors. So one of my collaborators has this model called the 4 core genotypes model, where you can look at the chromosomal contributions. So he's currently looking at the chromosomal contributions to cardiac fibrosis in the setting of valve disease. So probably, hopefully, I can put in a grant that includes both of those angles to fully interrogate everything. I think there's obviously other factors that contribute, epigenetic. I also want to go back and kind of combine the studies somewhat, so that, because a lot of the population of women have had a pregnancy, so doing the secondary stress later, it's thought that having a normal pregnancy and having normal lactation is protective. However, we'll be able to investigate that if we have both a normal pregnancy model and then also a comorbid model to see those mechanisms.
So in your model, it looks like if there was no lactation, it came back down,
Mhm.
correct? So, but if they liked it even when you took it to 1 year, it was still strong. Thing. So lactation seems to be a very critical part of the whole thing. So what happens during lactation?
So that's something we still need to look at a little bit more. But prolactin is known to have a hypertrophic impact, but there's also other lactogenic hormones that change during like lactation, such as oxytocin and everything, and that, I believe, that's also known to have an impact on the cardiovascular system. So we want to do some more studies to investigate the potential protective benefit of that lactation period, but also go back for those studies where we do age them out to have a non-lactation group there too. And then also make it even more complicated and do a similar thing with our multiple pregnancies study. But those studies are very complicated, so one step at a time.
So it's complex. Hydroxy urinary that is in the heart that is more important than what's the stem.
In terms of like
All the cardiac changes.
So it comes from the liver, and then it can be used by other organs too, I believe. And I believe there's also a small portion, I believe, that's produced in the kidney, but that's less.
Typically, and one other thing is tell them, you know, not to have too much ketones in the urine.
Mhm.
So because one of the ways that they try to avoid going on insulin is to fast overnight, and then the blood sugar in the morning will be better.
Mhm.
But it just bring up their ketone.
Uh,
So the question is, according to these studies, that's a good thing.
Yeah.
mhm.
Right? Not a bad thing. Because in the past, we always thought ketones was bad. You know?
Mhm.
It's not a good idea.
But it is. But the problem is in the setting of pregnancy, it's probably a double-edged sword because there's other publications out there that suggest that very high levels of ketone bodies during pregnancy can impact fetal growth and
That's the
development.
reason why we tell them not to.
So, yeah.
Tell them not to think because the fetal.
Yeah. So-
We don't want you, so we tell them to have.
I think you have to define which ketone bodies you're talking about.
Yeah.
We're not
Generally doing the
Mhm.
now, when we measure, we do the other color, even the strips that we have. They measure that.
Oh, well, that's done.
Yeah, we have, so when we did our mitochondrial studies, we also did provide acetoacetate. It didn't do anything.
Uh-huh.
But it's also unstable. So, that's a caveat to that. But there are specific ELISAs available for beta-hydroxybutyrate and acetoacetate, and we have done them. They do both increase during pregnancy. It's just that the free hydroxybutyrate or beta-hydroxybutyrate is significantly much more increased than acetoacetate. So we're assuming that most of the effects are from that, but we're not completely ruling out the impact of acetoacetate. We just have to do more experiments. Mm-hmm.
So can you have like a mouse model? After menopause or something in the female,
That
mice?
Yeah, that's the plan for future studies. So what we intend to do is there are a couple of different models. Obviously, no model is perfect because mice don't really have menopause. So we have to either completely remove hormones via ovarectomy, which is very drastic. It's like that. There's another model where you can inject VCD, and that is more of a perimenopause to menopause transition model. But so we maybe
Down.
So-
That's a question by Dr. Winter.
okay.
Major cause of maternal mortality, hypertension,
Mhm.
and considered examining hypertension rates.
So so the question was, have I considered looking at hypertension during pregnancy? Yes, I have a call tomorrow with a collaborator to get a preeclampsia model on board. I recently applied to the Gates Foundation to actually do a study utilizing 3 different preeclampsia models to do a extensive profiling for biomarkers. So we're trying to go after the RUP model, reduced uterine perfusion pressure model, the L-name model, and then there was one other model which was genetic. I think it was BPH5. So that it will take some time and some additional funding and such, but we'll keep on going. Thank
very much.
you.
I know
Thank you.
Have a time limit, so.